Great to see everybody here and have you all join us. This is the webinar series program for our Industrial Biotechnology Innovation Catalyst. I think everybody here probably knows who we are and what we do, but just in case, we are a Northwest-based catalyst activity funded through EPSRC as a place-based impact acceleration account program, and we're really here to help get the technology, get the science, get the knowledge that we have and hold within our higher education institutes across the northwest of England out into the wider community to generate that tangible impact and really help grow sort of the bioeconomy within our region. So we have grants available from 10 to 60k for relationship building, for doing some proof of concept project work, and also for secondments that are open at the moment. The next deadline is early June. And we also have a commercialization fund open at the moment, which is up to 50k in case anybody here fancies creating some tangible impact and looking to generate potentially their startup or spin out company from their own research. And that 50k can be used for most things, whether it be customer discovery, market advice, anything other than R&D to help along that translation journey. So I'll stop talking now. Really pleased that we have got Andrew and Ellie here today from the University of Liverpool, who are going to talk to us about the work that they do. So we're going to start with Andrew. So Andrew is a professor in physical sciences within the University of Liverpool. And he is a co-lead on our IBIC partnership that we co-created between Manchester, Liverpool, and our other institutions across the region. So, Andrew, I'm going to hand over to you, who's going to tell us all about enzyme design, and using it to create wealth from waste, I believe, today. So, Andrew, over to you.
Thanks, Ellie. Okay, so just going to share the slides. Okay. Right. Can you see that, everybody?
Perfect.
Yeah, good. Okay. Thanks, Celine. Okay, so it's welcome to this IBIC webinar. It's a great pleasure to have the chance to tell you about some of our work at Liverpool on developing enzyme biocatalysts for creating value-added chemicals from waste. So I'm going to start the talk, do the sort of first half of the talk, and then I'm going to hand over to Dr. Ellie Goulding. Ellie did her PhD in my group at Liverpool before moving to the MIB, where she is currently doing postdoc with Nigel Scrutton. Okay, so we'll crack on. So just by way of introduction, so we're generally interested in industrial biotech approaches for chemical synthesis. So development of an application of biocatalysts for chemical synthesis. So a couple of sort of key areas for us in the group, which I'm going to talk about, well, myself and Ellie are going to talk about today. So first one is bioplastics intermediates. So looking at conversion of bio-derived materials, bio-derived waste into intermediates for biodegradable plastics. And Ellie's going to talk about some of the enzyme conversions we've been developing towards sort of higher value compounds like pharmaceuticals by way of sort of demonstration. We're also currently active in the biobased surfactants area as well. In terms of process focus, I guess we're obviously interested in catalytic processes, given that we're using enzymes, so that's increased efficiency and in terms of time and energy, but also catalytic efficiency and selectivity. We're interested, as I said, in the bioconversion of non-food renewables, bio-derived renewables, to replace petrochemicals, in particular, in our case, cellulose-derived materials. And we're interested in reducing the use of organic solvents and toxic reagents, stoichiometric reagents, for example, for amide bond formation, which Ellie will talk about later, and also increasing efficiency by sort of telescoping or cascading mutually compatible reaction sequences. And so at the top of this slide, you can see just very generically, we're interested in, can we go from starting material A through intermediates B and C to product D with a cascade of enzymes, or even enzymes in combination with chemical catalysis, if that's compatible. And we'd be using the enzymes either in an isolated form, so we would call that in vitro, or as part of a whole cell system, so wholesale biocatalysis, or very often, if we want to scale up, look towards industrial processes using crude cell lysates containing the crude enzymes or cell-free extracts. So at Liverpool, we're fortunate in having all the requisite expertise, so obviously chemistry and biocatalysis and molecular biology, and then we have all the omics well catered for, and increasingly important, the computational biology and structural biology. So in terms of the way that we kind of approach enzyme discovery and development in our group, sort of broadly at Liverpool, we would typically start with literature lead, and then nowadays, I guess, looking at various methods, computational biology methods, in order to develop mutagenic strategies to move towards the desired attributes that we wish for the biocatalysts in terms of substrate selectivity, chiral selectivity, for example, but also biocatalyst stability and efficiency. And I'll say more about that as we go through. And then we're looking to make a selection based on often an iterative computational approach, and I'll say more about that later, to make a sensible selection for screening, and that would be as diverse as possible. Look at expressing those selected genes, and we do this within the Liverpool Gene Mill, so that's a facility associated with biosciences, where we're very fortunate enough to work with colleagues there to allow us to develop panels of enzymes for screening, and then sometimes we would iterate back through the sort of approach I've just described. So just in terms of the sort of project areas, past and present, so we've done a lot of work on cellulose conversion to bioplastics intermediates, and that involved identification and development of oxidase enzymes and methyltransferases. Then also very much an active area for us is the one in the middle in the blue box, enzymes for activation of carboxylic acids and conversion to amides, and that is looking at one of the most important reactions, amide bond formation in synthetic chemistry in all sectors, looking to replace the use of acid chlorides and or expensive rather atom inefficient amide coupling reagents, and Ellie's going to talk more about that. And then in the third box, degradation of plastics and valorization of the degradation intermediates to look at creating higher value products. And we've got some interesting examples of that. I'm not necessarily saying that they're going to be made into sort of industrial processes, but it's kind of proof of concept in most cases, I guess, in that area. So let's start off with the biomass conversion. So many of you will know this compound on the left here, HMF. This is can be made by degradation of cellulose via fructose, and we were interested in developing enzymatic approaches for the conversion of HMF into FDCA. Now, FDCA is a well-known drop-in replacement for terephthalic acid. Of course, terephthalic acid is a key component of polyethylene terephthalate plastic, PET plastic, but of course it's petrochemically derived, for the most part, at least. And so this FDCA is a bio-derived replacement, and is known to form plastics well, such as PEF and PBAF, which have good barrier properties and are bio-derived and in some cases biodegradable. So the key challenge for us at this when we started this project was this 6-electron oxidation from the aldehyde alcohol to the dicarboxylic acid using oxidase enzymes, oxygen-dependent enzymes using oxygen from the air, ideally in one-pot conversion from here to here. So it's kind of 3 oxidations, 3 2-electron oxidations to go from this material to this material. So it's quite a challenge. And to cut a very long story short, this is kind of where we got to. We actually went to higher concentration, but this was one of the graphs from one of the papers we published. So we found ourselves having to do this sequence in a one-pot, 2-step process, and I'll tell you the reason in just a moment. So here you can see the starting material. This is the cellulose-derived HNF. First enzyme is a galactose oxidase mutant M35. This was developed for different reasons in Nick Turner's group, and happily it turned out to be a rather good enzyme for catalyzing the conversion of the alcohol to the aldehyde. So this is the dialdehyde, and you can see the red line shows reasonably efficient conversion to the dialdehyde. We then add the second enzyme, which is an aldehyde oxidase enzyme, which then catalyzes the rapid subsequent oxidation reactions through to the dicarboxylic acid via the aldehyde acid. Both of these enzymes are metalloenzymes. The galactose oxidase is a copper-dependent enzyme, and this PAO-ABC is a molybdenum-dependent enzyme. So this slide really in the box in the center of the slide summarizes what I've just said on the previous slide. So here are the 2 enzymes that keep the main enzymes, galactose oxidase and the aldehyde oxidase, both using catalase to destroy the hydrogen peroxide, which is generated by reduction of oxygen as part of the process. And catalase, of course, very efficient, so that's not a problem. We're able to get very high yields of the FDCA. Now, we also looked at our ideal scenario, of course, was just to combine everything together, both enzymes with the catalase, with the air, to do the reaction in a single step, if you like. But the problem there was that the aldehyde oxidase would also oxidize the HMF starting material to the hydroxy acid. So it's oxidizing the aldehyde to the acid. And the problem there was that the hydroxy acid, this material, was a rather poor substrate for the galactose oxidase. So that was created a kind of bottleneck there, which was the reason for having to do the sequence in a stepwise fashion. And then we looked as part of this was done actually in conjunction with Bioplastics based near Southampton in the UK, with Innovate UK funding and BBSRC and EPSRC funding. So this was patented, and we got a few nice publications out of this. And we also looked at scaling this up with Professor John Blacker at the University of Leeds. But we did have a problem, actually, not with the scale up as much as the scale up of the production of the aldehyde oxidase. The cofactor biosynthesis is a bit of a headache and really still hasn't been properly understood to the point where one can produce large amounts of this enzyme on scale. Anyway, I'm going to sort of then move forward to the sort of next stage, in that we're having sorted the oxidation, we demonstrated the oxidation chemistry to the FDCA. We were then interested in activating these 2 carboxylate groups such that they could then render this material, this dimethyl ester, a substrate for polymerization. I'm not going to go into it today, but we'd also done quite a bit of work on converting this dimethyl ester into privileged intermediate actually for these synthesis of these bioplastics, which is the BDO, the bis butanediol ester, because BDO is butanediol is a bio diol, bio-derived. So that's sort of well established there. When we'd established we could use enzymes like PETase or related enzymes to convert the dimethyl ester into the BDO ester. So this was our challenge then, to how to dimethylate the diacid enzymatically, such that perhaps all these steps could be telescoped. And so we looked into the literature at enzymes that might do this. And this is a relatively unexplored area. An unexplored group of methyl transferase enzymes did exist. So they use this cofactor. This is called S-adenosylmethionine, or SAM. And SAM provides a methyl group from this position to methylate the substrate, shown by X here. And these molecules here, these methyl esters, are a number of naturally occurring esters which are produced in nature using these carboxyl methyl esterases. And we actually wrote a review summarizing the literature at this point during COVID. One enzyme that was attractive to us with this one called FTPM. This is an enzyme that was identified in the literature to be able to dimethylate this diacid here. This is fumaryl tyrosine, or fumaryl phenylalanine, depending on the R group here. But you can see the enzyme is able to doubly methylate both of the carboxylic acid groups. So we took this enzyme and expressed it and got reasonable quantities of the enzyme to allow us to try out some of these aromatic diacids that we were interested in, most notably the furan diacid that I showed you that was produced by the oxidation bioconversions. And the natural enzyme, the wild type enzyme as we'd call it, produced a mixture of the dimethyl ester and also the monomethyl ester, where just one of the carboxylates had been methylated. Interestingly, it also worked on terephthalic acid, similar scenario, and other dicarboxylic acids, including this pyridine one, which can be made from lignin, and Tim Bugg's group at Warwick have done a lot of work in that area. And also this acyclic molecule, interestingly, was very selectively monomethylated. So that looked promising. However, when we looked at these reactions in more detail, what we found, looking at the time course, so this is the disappearance in red of the diacid and the production of the monoester in blue, and then the diester, which is the polymer precursor, in green. You can see very or reasonably rapid conversion to the monomethyl ester for the furyl system, but rather sluggish conversion to the diester. And for the terephthalate, this is even more pronounced. So the very rapid conversion to the monomethyl ester, but rather slow for the second methylation to produce the diester, the polymer precursor. So we had a look at this enzyme. We made a model working with Professor Dan Rigdon at Liverpool, who's my colleague that we collaborate with extensively, who's an expert in structural bioinformatics. And when we looked at the model, we docked in these substrates. So this is actually a superimposition of the furan and the terephthalate diacid. We could see that there are two residues here, are arginine 27 and glutamine 31. They're responsible for positioning the carboxylic acid group to be methylated by the cofactor. Here's the methyl ready to be transferred, and also some other arginine residues on the other side of the active site, defining a strong positive charge in the substrate binding pocket. So we decided that if we could perhaps mutate the arginine 166 to a less polar residue, we might encourage the substrate. Once the monomethyl ester has been formed, it presumably has to leave the active site and revisit, or somehow turn around to present the other carboxylic acid group to the SAM cofactor, another molecule of the SAM cofactor. And so the existing monomethyl ester has then to position itself in this area. So we thought perhaps getting rid of the charge there might be helpful. And indeed, that turned out to be the case. So this slide shows some mutants that we made at the position arginine 166. This is the arginine side chain. You can see it's quite polar, good for binding carboxylic acid groups, but less good if you've got a less polar monomethyl ester having to sit in its vicinity. And to cut a long story short here, you can see here's the wild type enzyme producing, as we saw before, a mixture of the diester and the monomethyl ester here, with a small amount of the diacid remaining. Here's one of our best mutants, the methionine mutant. So this is an apolar side chain, giving much more conversion to the dimethyl ester, and interestingly, a less efficient conversion of the original diacid. So that kind of fits with our hypothesis. And indeed, when we incubated the monomethyl ester as a substrate, rather than the diacid, we see a number of these mutants doing a very highly efficient conversion to the dimethyl ester compared with the monomethyl ester. You see these high numbers here. And we also tried terephthalic acid, so that's the benzene dicarboxylic acid. And there, it's similar sort of scenario with the wild type enzyme. We get a mixture of dimethyl and slightly more of the monomethyl, and then we can reverse that when we put a apolar side chain phenylalanine in place of the arginine 166. And again, when we incubate the monomethyl ester, we see much higher conversion to the dimethyl ester. So that was very pleasing. That was Lucy Ward, actually, who did that work. Lucy's recently completed a postdoc in the MIB with Nick Turner, and I believe now has moved to AstraZeneca. So the top of this slide really just summarizes then the combination of the oxidation chemistry to get to the dicarboxylic acid from the bio-derived HMF with the mutant, the R166M mutant, to enable us to get to efficient conversion through to the dimethyl ester. And there you can see the chart showing the conversion now to the dimethyl ester in a good time scale compared with the wild type enzyme. So we were able to, through a fairly simple sort of rational mutagenesis strategy, able to improve things considerably in terms of substrate selectivity. So we've also been looking at recycling. Can we break down plastics and recycle them back to their monomers, or then use their monomers to synthesize higher value intermediates? So one of the things we demonstrated was we got hold of some polyethylene furanoate. So this is the PET analog with the furan core, and we were able to use an enzyme from the literature, cutinase enzyme, to produce the acid, and then use our mutant methyltransferase to generate the polymer precursor, which could be then used to make these bioplastics. And I won't say too much about this, but Ellie's going to talk about how we were able to use milled waste PET plastic and then use hydrolases and in some cases methyltransferases to make activated esters. And then Ellie's going to focus on the use of promiscuous acyltransferase enzymes, which are basically hydrolases, but able to work in water and catalyze the amidation of some of these monomer intermediates towards higher value compounds. I just wanted to mention something else that we've recently been quite excited about at Liverpool, working with Dan Rigdon in biology, in computational biology, and this is Margot Hone is doing this work. She's a PhD student in my group, co-supervised by Dan, and Margot has been developing a pipeline, a computational pipeline, to allow us to predict multiple mutations to improve soluble expression of enzymes. So one of the bottlenecks, if you work in this area, often is the ability to produce sufficient quantities of enzyme for even to do your research, but also, but obviously importantly for scale up. So we're taking a sort of new approach here by combining a number of algorithms in order to try to make predictions about multiple surface mutations in order to improve soluble expression levels and yield of active enzyme. So just briefly, I'll run through this. We're using something called AgriScan to identify surface residues. We give them a non-zero score, so they're polar residues, excluding binding pocket residues. We then use something called soluble MPNN to design, in this case, 1,000 new sequences for this particular enzyme example. And we test those sequences in a number of ways. We give those sequences to another algorithm called protein MPNN, which tests the probability of getting a properly folded and functional protein. So these are trained on large databases, not developed by us, but these are open access, openly available algorithms to use. We then make AlphaFold structures of these sequences and test those in terms of their stability compared with the wild type, the original enzyme, and also the global solubility score prediction. And we take those three factors together to come up with a short list of enzymes to then express in the lab. So we took 14 in this case, and then 3 of those turned out to have much better soluble expression, and importantly retain their activity and selectivity. And that then of course can be iterated to improve things further. So we're actually applying this to a number of the enzymes that we're working with in the lab at the moment. And this appears to be quite a promising approach beyond the sort of traditional approaches that one can obviously take to improve enzyme expression. Just want to mention the computational biology facility at Liverpool. This is part of the shared research facilities, including the gene mill, which are able to offer partnerships or services in a number of different ways. So with historic strengths in training and workshops in omics, data analysis, machine learning and databases, but more recently expanding into protein bioinformatics and all of these areas listed here. So it's possible to contact this group and work in a number of different ways, including inclusion of time on grant applications or if you're interested, please do use this QR code or make contact through myself if you're interested in pursuing this with the group at Liverpool. Okay, so I just, before I go on to thank a few key people. So Lucy, I mentioned, did a lot of the work on the methyltransferase development, and Ellie's obviously going to tell you about her exciting work from her PhD. Faye Allen is currently in the group and doing great things with some of the other enzymes we're developing for the acyltransferase. And I mentioned Margot been developing this novel bioinformatics pipeline. And then a few key collaborators I mentioned, Dan Rigdon, also Enrique Salcedo-Sora. So Enrique manages the gene mill at Liverpool. This is set up with BBSRC funding, but then also expanded and supported by the university. So that's been a really great facility for us, and the collaboration with Enrique as well has been very productive. And I'd also like to acknowledge some of our key industrial partners, so Biome Bioplastics in particular for this work, and also Prosomics for supplying some of the enzymes and also some of these the funding bodies shown here. So I'm going to stop there and hand over to Ellie.
Thank you. I'll just quickly share my slides. Can everyone see those okay? Yeah.
Yep.
Well, thank you. So hi, I'm Ellie, as Andrew said. I completed my PhD last year in his group. And since then, I've been completing a postdoc here at the University of Manchester within FBRH. So today I'm going to be talking through some of the work we did at Liverpool, which focused on the applications of acyl transferase enzymes for amide synthesis under aqueous conditions. In particular, we looked at the development of enzyme cascades and protein engineering to allow sustainable amide production. So just a bit of introduction. One of the key challenges for organic chemists has been listed as sustainable amide synthesis. So there has been increasing interest in biocatalytic methods to try and address this issue. They do have the advantage of mild reaction conditions and improved selectivity by avoiding toxic organic solvents and protection or deprotection steps. We therefore aim to develop a biocatalytic system whereby amides could be formed from carboxylic acid via an ester intermediate. And this is using methyltransferase and acyl transferase enzymes, and that is all under aqueous conditions. So I'll talk a bit more about this cascade in more detail later on. But my project was mainly focused on the development of enzymes for the acyl transfer step, so I'll mostly talk about these efforts. Specifically, one of the key aims for us was the use of PET monomer terephthalic acid as a starting material, which can be repurposed to valuable amides. The valorization of plastic waste is an emerging area of research, so upcycling of PET is of quite significant interest, both academically and industrially as well. So we know amides are valuable compounds used in pharmaceuticals and other industrial sectors. In terms of being formed enzymatically, mainly ATP-dependent enzymes have been explored, but they can be quite costly. So hydrolyses have therefore generated a bit of interest as they don't require any cofactors. The way they work is by catalyzing the acyl transfer from esters, either alcohols or amines, to form esters or amides, respectively. So they usually have to be done in organic solvents to overcome the competing hydrolysis of the starting material or of the product to the corresponding carboxylic acid. More recently, some hydrolyses have been found to have a natural kinetic preference for acyl transfer over hydrolysis. So the acylated product is formed much faster and can accumulate at a higher concentration before hydrolysis has the chance to occur. And this is in bulk water as well, so these are called promiscuous acyl transferases. Conducting these reactions in water is much more favourable to improve sustainability, efficiency, and it also allows compatibility with other enzymes for enzyme cascades as well. So acyl transferases are quite an unexplored class of enzymes with this unexpected reaction promiscuity. So we were particularly interested in utilising this for amide formation. Specifically, the main enzyme we focused on was PEST-E, which is an aromatic esterase reported to have acyl transferase activity for amide formation by Bornschau's group in Germany. It was part of a screen which found enzymes with more hydrophobic active sites had generally better acyl transferase activity in water. So PEST-E, which is shown on the top line on the graph, was one of the key hits that had a kinetic preference for acyl transfer over hydrolysis, and it has since been used in further studies to form other esters and compounds with direct applications as well. So we chose to focus on this enzyme, and we found it was also able to catalyse amide formation in buffer. So using methylfurate ester and benzylamine as model substrates, under the optimum conditions, which were mild conditions of 25 degrees, pH 9 buffer, up to 93% conversion to the amide could be achieved in 6 hours. That's the blue line on the graph there. There was also minimal hydrolysis, shown by the grey line, which is mostly due to spontaneous non-enzymatic hydrolysis of the starting ester anyway. So that just shows the natural kinetic preference there. A preference for amidation was also seen when we tested several ratios of ester and amine. So even when a twofold excess of ester was used, there's still 60% amide formed, which just really highlights this natural acyl transferase activity of pest A. We screened a range of amines to form different furan amides, which can be of industrial interest due to their use in polymer and agricultural industries, and some of them also have antiviral and anti-cancer properties as well. PESTI accepted a broad range of benzyl amines when coupled with the methyl furorate ester, with different functional groups being tolerated at different positions on the ring. So methoxy, nitro, cyano groups in particular were tolerated particularly well, maintaining the high conversions as with the parent amine. More challenging amines like anilines and secondary amines were also accepted. This was particularly interesting as the lower nucleophilicity of anilines and the steric hindrance of secondary amines mean that they're not usually accessible by these types of enzymes. And these substrates are more challenging and are a bit more synthetic relevant. So that was quite promising as well. PEST-E can also catalyse amide formation with a range of acyl donors in quite good yields. So usually only simple acetyl esters are used with these types of enzymes in quite a large excess to drive the reaction. So these larger amides formed from bulkier, less soluble esters are more synthetically relevant, and they also have the potential to be used in final products or intermediates, especially those like with dependent groups like a nitro or acid group, as these can allow for further functionalization to form more complex compounds. So they also showed varying amounts of ester hydrolysis as well, which indicates that this is quite substrate specific and dependent on the amine preference or reactivity. So leading on from these results, another key area of interest for us was incorporating this reaction and the methyl transferase reaction that Andrew talked about earlier into a one-pot enzymatic cascade with the aim to form valuable amides from waste plastic. So 400 million tonnes of waste plastic are produced globally each year. So upcycling post-consumer PET from plastic bottles is quite an important and attractive concept. Just to briefly go through the cascade, it starts with waste PET from a plastic bottle being milled down and then hydrolyzed to its monomers terephthalic acid and ethylene glycol, and that was using a quadruple mutant of ketonase LCC. So terephthalic acid can then be monomethylated by methyltransferase FTPM to form the monomethyl ester, which then has the potential to be amidated by PEST-E. So we identified this tamibarotene as a key amide target, as it's an amide that has pharmaceutical properties, as it's been shown to have antineoplastic activity against acute leukemia. So the first two steps of the cascade can be achieved quite efficiently, with 5 mg of waste PET being converted to 1.5 millimolar of the monomethyl terephthalate, with the second step achieving 93% conversion. As a separate step, starting from terephthalic acid, the FTPM reaction can be coupled with PEST-E to form tamibarotene in one pot. So this really demonstrates the proof of concept that PET monomers like terephthalic acid can be valorized to high-value amides like this anti-cancer agent. So the system was also applied to some other aromatic amides by substituting this amine for a few different ones shown in the table. And this really showcases the potential of this to be utilised for broader amide formation as well, not just one particular target. So we showed the full 3-step enzymatic cascade from PET can be used to transform waste plastic bottles into the benzyl monoamide here, using benzylamine as the acyl acceptor for the last step. In relation to this, we also looked at an alternative route, so route 2 shown in blue here. This would avoid the cofactors needed for the methyl transfer step and also reduce the overall number of steps in the cascade. So the way this works is that by using petases to hydrolyze PET instead of the ketonase, the intermediate MHTT can be accessed, and this can then be utilized as a substrate for amidation by a cell transfer. We were pleased to find that PET-E can catalyze the coupling of MHTT with several amines to form the various monoamides, and these yields were comparable to when using monomethyl terephthalate as the acell donor. This again just highlights another potential route to repurposing plastic waste to more valuable compounds, and doing that in quite a sustainable way. Another aspect I wanted to talk about is we looked at using mutagenesis to improve the amide bond forming activity of PET-E, and also to reduce the unwanted hydrolysis activity. So really trying to look at how to harness and refine this kinetic preference for acell transfer to allow more efficient reactions. We did this by structure-based rational design and used the formation of tamibarotene as a target because of its pharmaceutical applications. We performed substrate docking of the tamibarotene amine in the active site of PET-E, so the amine shown in pink on these figures, and we then looked at identifying possible nearby residues for mutation, which are shown in yellow. So based on previous reports of acell transferase mutagenesis, we categorized the mutants into 4 main strategies for improving acell transfer activity over hydrolysis. So briefly, they included increasing the size of the acyl acceptor tunnel to accommodate bulkier amine substrates by mutating larger residues like phenylalanine to smaller ones. Another important strategy was to disrupt the water network. So what we mean by that is mutating residues that bind to water and strengthen its position in the active site. So removing this interaction would disfavor water from the active site and therefore reduce hydrolysis. So we looked at increasing hydrophobicity as well by just mutating any hydrophilic residues to make them a bit more hydrophobic. And finally, we looked at a less studied strategy, which was mutating residues that help stabilize the acell enzyme intermediate. So the thought process is that as amines are more nucleophilic than water, only they will be able to, or more, be able to attack the acell enzyme intermediate more. So some mutants do overlap into other categories, but this generally was the basis of where we chose the targets for mutagenesis. So 5 single point mutations were initially tested with the model reaction, which was the methylfluoroester ester and benzylamine, shown on the left. And we also tested the tamibarotene reaction, which is shown on the right there. So with the model reaction, it was found that 2 mutants, F289A and F33L, had improved activity compared to the wild type. So interestingly, they both used the strategy of increasing the size of the acyl acceptor binding tunnel. So that indicates that was really quite an important strategy, if not the most important strategy for improving acell transferase activity. So for the tamibarotene reaction, the F33L mutant showed a 4-fold increase in amide conversion compared to the wild type. So this significant change could be attributed to the sterically demanding amine having better access to the active site, which was allowed by the bigger tunnel that we generated with the F33L mutant. So with these 2 key hits in mind, we did some double mutants based on these results, and also another single mutant, F33A, which would create even more space in the amine binding tunnel. So the mutant that combined the 2 key hits from the first round, F33L and F289A, had the highest activity of the mutants in the second round with the model reaction. And this was 1.2-fold higher than the wild type, but not necessarily significantly higher than either of the 2 single mutants individually, which suggests that combining these mutants had no particular effect, at least for the final amide conversion. So no improvements were seen with the tamibarotene reaction with any of the double mutants, which suggests a limit to the mutation effects, and in some cases it's not quite enough to overcome the substrate limitations. However, the F33A did maintain the high conversion seen from F33L, so there was no loss of activity there, and that further reiterates that the size of the binding tunnel is quite important for reactivity. So just to look at the activity of the key mutants more closely, time courses revealed that F33L and the double mutant had higher rates for amide formation compared to the wild type. So the blue lines are amide formation on the graphs there. This was even using equimolar amounts of ester and amine, so really quite significant activity. The hydrolysis, shown in grey, is more significantly reduced with the double mutant on the right, which suggests a synergistic effect of the two mutants to allow a much faster kinetic accumulation of the amide. So this proves that it is possible to manipulate the activity of PEST-E to reduce the competing hydrolysis of the ester, and also that this strategy can be applied to other acyl transferases as well. We also scaled up the reaction, so the model reaction and the tamibarotene reaction as well, to demonstrate that high-value amides can be produced in this way, and they didn't require a purification step as well, which further increases the sustainability and scalability too. So yeah, I'll leave it there. I just want to thank Andrew for his support with the project, in particular Lucy Ward and Faye Allen for their contributions as well to the project with FTPM and the scale-ups, as well as the rest of the Carnell group and GMIL as well. Thank you.
Fabulous. Thank you very much, Andrew and Ellie, for that excellent summary of exciting activities happening around the design of the biocatalysis for different transformations. So we do have a couple of questions in the chat, actually, and if you've got any more here this afternoon, please either pop them in the chat or pop your hand up, or just pop your camera on and shout out. We'll come to you. But the first question is from Francesco. Andrew, presumably recycled plastics streams have really tight cost targets. How realistic do you see an application of methyl transferases in this space, given the significant challenges associated with the SAM? I think.
Yes. Thank you. Yeah, thanks for that question. Yeah, there are recycling methods around, as you probably know, for methyl transferases. We didn't actually use any of those. We were more interested in demonstrating that, establishing the methyl transferase activity. But yes, it's a good point. I think there are opportunities for wholesale recycling. So one thing we have tried but not had much success is co-expressing the methyl transferases with other enzymes for the cofactor recycling. But it's not been very efficient. So that's one of the reasons we're actually very interested now in looking at the other esters, the esters that can be derived by direct breakdown of the polymers like PET. So the hydroxy ethyl monoester is, of course, what one of the main products you get with wild type PETase, and we're very pleased to see that works with the acyl transferases. So that would cut out the methyl transferase. That's not to detract from the methyl transferases are interesting, perhaps for, but more for higher value products like pharmaceuticals. Perfect.
Thank you very much. Another question from Natalie. This might be for both of you. And it's again about thinking about that broader industrial adoption and what challenges do you still see, maybe apart from the cost, before we can get these types of things out more widely?
Yeah, I can start and answer that, and Ellie may want to join in. So I think obviously we've mentioned that the cofactor SAM with the methyl transferase issue. I think one of the things we're most excited about is these promiscuous acyl transferases. That they're basically hydrolase enzymes that give the kinetic acyl transfer reaction in some cases. So we're looking to expand the range of substrates, and we've got a number of different enzymes that work on different ranges of substrate, but of course they're not cofactor dependent, and we've already shown actually that we can produce these enzymes on reasonable scale. And I think, you know, it's well known that lipases and esterases are scalable, and you know, if we can build into that the in situ product recovery by, you know, dropping the product out of solution, as this seems to be the case with some of the reactions, then that looks very good for scale up. Ellie, do you want to comment at all?
Yeah, just similar in that we were thinking about this with the mutagenesis, really trying to find a way to, you know, reduce enzyme loading for scale ups and also be able to use higher substrate loadings as well. So we did kind of do that with the scale up, where we managed to get it up to, I think it was 50 millimolar of ester as a starting material. So obviously that's not industrial scale, but yeah, it's just thinking about that early on and trying to find mutants, I think, that would be able to be a bit more scalable. Yeah, it's kind of like the focus and what's kind of stopping it from being industrial scale.
Yeah, I should add actually, we've got enzymes now, one particular enzyme in the lab, which is about 1 million times faster on the model reaction than PESD. So that's quite exciting.
Wow, that's quite a step change indeed. Any final question from anybody? Anybody brave enough to come on camera or pop your hand up? No, then I'll ask one before we finish. Oh, it's just kind of building on the questions around sort of the industrial adoption, and you know, one aspect is this scalability of the enzyme production, and then the product purification or separation, I guess. But thinking about the sort of, I guess, the next step in the scalability, what would you need to do to kind of prove that this would work on the pilot scale? Have you thought about that at all in terms of engaging with a company that is recycling plastics or that type of thing?
Yeah, so we've got another project which I can't really talk about too much, but we are looking at this. So we're kind of designing the reaction conditions with very much with the product isolation in mind, and scalability, substrate concentrations, solubility of the product, and the solvent composition that we'll need in order to drive the reaction. So render this kinetic acylation, for example, irreversible, capturing the product by in situ precipitation, and then how we then recover the product and make the whole thing more sustainable and efficient in terms of the throughput. So yeah, that's very much in our sights. We also obviously there's the issue with the methyl transferases, but we've also got some work now where we're getting acid plus amine rather than ester plus amine. There are also, you know, in order to drive that, you have to drop out the product as it's formed, but that is also working. So that's another exciting area for us.
Very exciting, actually. And I'll leave the last question, which is on a similar vein to Kirk. And he knows that you were just doing a demonstration reaction, but I think it shows the level of excitement in the technology. So do you foresee any regulatory problems using waste material as pharma feedstocks? You can answer yes or no.
Don't know, possibly. We haven't really thought about that, but it's good. It's a good point. Thanks, Kurt.
Perfect question. and one that could be debated potentially when we meet again, hopefully at our conference, which will be on the 10th and 11th of June over in the Spine in Liverpool. So watch LinkedIn space and your emails for more information on that in the coming few days. So with that, just want to thank Ellie and Andrew again for a great seminar today. Our next session, next online webinar is in May. We're going to miss April because it's Easter. So 15th of May, we've got Andrew Dean and Eddie Banian from Manchester Metropolitan University who are going to join us and tell us a little bit about the work that they're doing over there. So thanks very much. Lovely to see everybody, and look forward to seeing you all again soon in May, if not before. Thanks.
Thank you. Thanks, everyone.
Thank you.
Bye.
Bye.